Welcome to the MS-Perspektive podcast, Perspective with a K as I'm German. I'm Nele van Horsten, MS patient, patient advocate and author. I'm bringing you interviews, inspiration and information about living with multiple sclerosis and show you ways how you can positively influence the disease and make the best out of your diagnosis. On ms-perspektive.com you will find the show notes, 11 free tips as a PDF and lots of other information. And now the show begins. Thanks for tuning in again and welcome to episode number 103. Can epigenetic research transform MS treatment? Insights into DNA methylation and MS progression with Dr. Majid Pahlevan Kakhki. Enjoy the interview. Hello, Majid. It's such a pleasure to have you on the show. And hello to Stockholm in Sweden.
Yes, hello. Thanks for having me here. It's a great pleasure actually to be with you. And finally we are here.
Yes, great. And before we actually start with the interview, it would be lovely if you could introduce yourself to the audience so they know who is with me on the podcast today.
Sure. I'm saying hello to all your audience as well. And my name is Majid Pahlevan Kakhki. I'm a doctor in molecular genetics, working as a researcher in Karolinska Hospital in Sweden, as you mentioned. And I'm originally from Iran, and from the northeast of Iran, where we are cultivating a lot of saffron. So saffron is coming from our region, and I think it's an antioxidant that people says that it's beneficial for MS patients as well.
And it looks good. And yes, it's always lovely to have the yellow rice.
Yes, of course.
Okay, so what was your motivation for your career choice?
I mean, that's a great question. I really love that question every time that I hear about it. So the thing is that from my childhood, I was always eager to learn something new and to have some innovation. So that was something that actually motivated me to go to something that has all these kind of criteria. And there were a lot of other things. As I mentioned, saffron, my whole childhood was working on farms with my parents, and I was smelling always like saffron. It's a good smell, I should say. But the thing is that at that time, it was a little bit tough for me to have enough financial support for my studies. So actually, I became a tailor first. I was going to the shops and I was doing tailoring for a while. And one of the times in this shop when I was tailoring, there was a radio on, and the radio was talking about some research that researcher has done on the brain of some animals, and they have seen some changes. And that was actually super interesting to me. And at the same time, my mom was telling me always that you should go and study something, because at the end, in agriculture and cultivating and then doing this agriculture is not an easy work. Mm-hmm. And then I said, okay, why not doing some research? And I will go to that one. Then I followed this way, and now I'm here with you.
Fantastic. And I always love it when people are doing, let's say, like normal work, because then they can appreciate everybody's else jobs. And I mean, when you are just going straight to university and never touched, let's say, the working with hands business, I think it's a different experience. It doesn't have to be bad, but it's a different experience. And I was doing like a graphic design before, and then decided to go to university. So I, on my own, had the same kind of experience. Of course, not agriculture, but let's say moving on to different fields. So before we dive deeper into epigenetics, I would like to have a slight introduction. So could you explain what epigenetics is and how it differs from genetics in relation to multiple sclerosis?
Yes, sure. I think it's a very good start. So genetics, actually, when we are talking about genetics, we are talking about DNA. And DNA is the sequences that we got from our parents, and it's actually shaping our body somehow. Whenever we are talking about genetics, we are talking about those chromosomes that are coming from the dad and mom, and then normally they are remaining as it is throughout our life. So they are not changing a lot. But epigenetics is another term. So we know that with our defined genetics from our parents, we are always exposed to some other factors that they are coming from environmental factors, actually, we call them. So these factors can affect the genetic regulation. So it means that when some environmental factors are coming to us, it can change the way that genetics expresses itself. So here is the term of epigenetics coming. So epigenetics are those factors that can regulate the genes, but without changing the sequence of the genes. So it means that there are some factors coming from the environment. They can change the gene expression or the regulation of the genes, but at the same time, they are not changing the gene. So I can make, as an example, it's like a switch that is turning the gene on and off. So we call these things as epigenetics. And in terms of multiple sclerosis, we know that as a complex disease, MS is both actually made maybe from genetics and also environmental factors. And whenever we are going to see how environmental factors affecting genetics, and then a person is susceptible to MS, we see the term of epigenetics. And so that's how it is. And we know that there are many environmental factors that they are important in terms of making someone making someone susceptible to MS, or maybe make someone more progressive with a more progressive form of the disease. So it's happening in that way. Yeah. And I think epigenetics is one of those factors that can affect both of these situations.
So kind of, to give it a picture, kind of the board, the keyboard of the DJ, of the disc jockey, volume up, volume down, and
exactly,
kind of
exactly. That's a perfect example. Yes. Yeah.
So what is DNA methylation, another word, and what role does it play in regulating genes that might influence the progression of MS?
Yes, we have a lot of epigenetic factors when we are calling them epigenetic factors. There are a lot of different examples of them, and DNA methylation is one of them that is very, very much studied. So and there are some key features of this DNA methylation that it makes it suitable to study it. Like, first of all, DNA methylation is something that happens to the DNA. So it means that there is some specific part of the DNA that, for example, is getting methylated, and methyl is a kind of a chemical changes that is happening to the DNA. So it's not changing DNA, but it's sitting on DNA, and then that's what we call it DNA methylation. And this DNA methylation has three specific features that make it very ideal for this purpose. So first of all, it's sensitive. And and whatever environmental factor is coming to us, actually this DNA methylation can recognize that one and can change because of that environmental factor. So we call it it's a sensitive factor. Mm-hmm. The other thing is that it's reversible, and this is like really nice. So when it's sitting somewhere and it's sensitive and sitting somewhere recognizing one factor, then we have also the chance to reverse it to the normal situation. And that's the opportunity that gives us, maybe in future, we can make some medications based on that. And the third one is that it is stable at the same time. So if something happens and nothing is actually reversed to the normal one, still we can track it back. Like, for example, if someone is smoking, there are specific marks happening, like adding to the genome as DNA methylation. And after 10 years, even still, we can recognize that these people has been exposed to the smoking factors or not. And that's how DNA methylation is important. And most of the time, it's nice for us because we can use it as a biomarker. And nowadays, biomarker research in MS is something super important because we want to understand in advance that if someone is progressing like in MS, or is someone like they're responding to a medication or something like that. But in terms of multiple sclerosis, we have had a lot of research and showed that, okay, whenever we have a genetic background of MS, it doesn't mean that everybody will get MS. So we need to have a specific setting on the genes that genes and plus the environmental factors together can make someone susceptible to have the disease or to show the disease. So in this situation, DNA methylation is one of those factors that shows that, okay, now we have this genetic background. And if you have this kind of like epigenetic setting, then at the end, the consequence will be to show to show the symptoms of the MS.
Yeah, great. Yeah, and there is very interesting research on identical twins where not both are having MS. I had somebody, Professor Dr. Gerdes, on the German podcast. I have to invite her for the English one, but it's very interesting. Luckily, not all identical twins. The second one is getting MS, so that is a lot of epigenetic happening there. Exactly. So let's start to dive into the topic. How do genetic variations at the 1q21.1 locus, nobody has to remember that, contribute to the risk and progression of primary progressive MS?
Yes, that's a great question. And I think you are referring to my last paper that we published in Nature Communications. So just to let you know, 1q21.1 is an address for the gene. So normally we are marking the genes and chromosomes like that. So that's simply like as an address to the genome. And then we are referring with that number to a specific site of chromosome 1, where we found that there are some genetic or epigenetic factors that they are linked to a primary progressive form of MS. So as a background, we know that we have at least three different forms of MS. We have relapsing remitting, that is like more than 85% of MS patients starting with this. And then after maybe so many years, some of them are progressing to the secondary progressive. But the primary progressive is just 10 to 15% of MS cases. So they are not many, but they are very progressive from the beginning. And the thing is that this specific situation actually gave us this chance to understand why actually there is a difference between the people in terms of relapsing remitting and primary progressive. So progression is coming from where? And we have started with DNA methylation because we were thinking, okay, with the relapse remitting, there are lots of studies, but with primary progressive, we have still very few. So with that, we understood that, okay, there is a specific region on chromosome 1 that is actually has more methylation, as we discussed before. So this more methylation makes these people more susceptible to go for primary progressive, to have like more progressive MS. And that's how the project started. And what we have done a lot of analysis and a lot of kind of different experiments to understand how this link of genetics and epigenetics together can make someone more susceptible to MS. And we understood that, this can affect actually the neurons in the brain. So it means that whenever we have these marks in our genome, then it can make the neurons in the brain somehow more susceptible to not work properly. And then that impaired working, actually, it's making the person more susceptible to MS. And one important thing here is that we found is that we first did the analysis on the blood of MS patients. But we know always like MS is an autoimmune disease. So there is a link between the blood and brain. And the other important thing that we found is actually that there is always a link between the blood and the brain. So we know that the immune system from the blood actually is entering the brain and then doing a lot of job there. And then at the end, people shows the symptoms of the MS. So here actually we found that a specific link between genetic and epigenetic at that specific site of the chromosome 1 can make someone susceptible for more progression in terms of MS.
Okay, great. And it's really great that you are doing research on the primary progressive, because I always hear from the community that they are very sad that not so much research is done for them. And I think it's really starting to kick off now more and more as we have a lot of good treatments for the to prevent relapses and MRI activity. So how do specific genes like CHD1L and PRKABW2 influence the progression of MS? And again, nobody has to remember that one, but...
Sure. Again, to explain that in that specific address that I just mentioned, there are many genes actually located there. And the two that you mentioned is those genes that they are located in that specific address. So they are living in that address that we just mentioned. And just to explain that we know that in our genome, we have more than 20,000 genes, so in different chromosomes, and they are working together. And these works together actually makes our body and then all the functions that we do have. But we do not know what genes exactly do. So I'm still after 20 years that we have still, it's more than 20 years that we have the genome sequence of the human. So we know all the genes are in the human body. But still we don't know what exact function they do have and how they are working together. And these regulations are super important in terms of knowing the mechanisms behind the diseases. And one example was this CHD1L. It's a gene that is not very well known that what it is doing. We know that it's actually sometimes helping to repair, for example, the DNA, but not always. And it's not the only job that it's doing. But the beauty of this work is that actually CHD1L was already known that can affect the people with autism and schizophrenia and even some mental retardation. So that was interesting that actually there is a link between this gene and the brain function. But nobody actually claimed that one for MS. So we have tried to understand that link. And as I discussed, we showed that, okay, there is this genetic background and epigenetic regulation, and then we linked it to the function of the neurons. But at the end, to understand really how this is working, we researchers, we are trying to make some models. And based on that models and then manipulating this gene, we can understand how is the function of that gene. So in our case, we made a model of zebrafish. It's a small type of fish that we are using in our research. And then we try to modulate, to regulate a little bit in different way, this CHD1L gene that is located in that address that I mentioned before. And then we realize that when we reduce the expression of these genes, then we see that the neurons are functioning differently. So they are not doing a normal function. So it means that this gene is important in the function of the neurons. And at the same time, to understand, okay, zebrafish is not a human. So there would be another genes there would be another regulations, even environmental factors are different, a lot of other things. To make a more kind of humanized model, we used iPSCs. So these iPSCs is part of like stem cells. Let's just call it the stem cells. So these stem cells, we are taking from the patients, and then we are making these stem cells from the blood or from the fibroblasts in the skin. And with the help of them, we can understand that, okay, because these stem cells are coming from the patients, they have the same genetic background as the patients. Now we are changing the genes, and then we understand what is happening. And then we have done this job, and we understood that CHD1L and other genes around, they are very much involved in the neural function. So with changing this gene, we understood that these iPSCs, these neurons that we made from these stem cells are not functioning normally. And now still this research is ongoing to the next level, and we are trying actually to understand how we can have a better control on these genes.
Fantastic. And let's look a bit into the future. In what ways could these findings on DNA methylation and gene expression potentially lead to better understanding or treatment options for MS?
Yeah, I love this question because it's actually something about the future that we are trying to get there. And going back to the RRMS situation and PPMS situation, so we know that for RRMS, nowadays there are a lot of options out there in the market as medications. Because in the RRMS phase, we have a lot of inflammation going in the body. And we have a lot of nowadays medications that can actually control this inflammation and then suppress the disease and suppress the symptoms. But while for the PPMS, as you mentioned also before, there is not much actually options available. So with this situation, we think that one of the main reasons that makes PPMS a little bit difficult to make medication is that whatever is happening on PPMS is about progression, and progression is normally happening in the brain. And the brain is somewhere in our body that is well protected. It's not easy actually to access the brain. And that's why we have very less knowledge from the brain than peripheral blood, for example, or immune system. So in this situation, we were thinking, okay, now we need some specific type of research that actually show us how we can deal with the complexity of the brain when we put together genetics and epigenetics factors. So we know that the link between genetic and epigenetic factor is affected by the environmental factor, but how these environmental factors actually are affecting the brain function, that's something very important. So we are trying nowadays by integrating genetic, epigenetic, and molecular data together to move closer to identify key regulatory pathways that they are involved in MS. This is something very, very much in the future that we want to know. So we want to know, when we have a specific genetic background is defined already, which epigenetic factor means which environmental factor can affect that, and then go to the progression of MS. And this is something very interesting, because it's actually this integrating of different layers of data together can actually help us to find better biomarker for disease progression. Mm-hmm. So we don't want to wait for a patient to be progressed in MS symptoms and then we do something. We want already from a specific type of biomarkers that we will have to already know that, okay, now progression starts, and then we can offer like these kind of medications. And also another important thing that can help us in future to better control MS is actually to have personalized treatment. So with this personalized treatment, we don't want actually to have a medication like for all MS patients. We want a specific medication based on the genetic and epigenetic signature of that specific person that works 100% for that person, not working even 50%. So that's how we want. And then we know that in MS, we have a lot of heterogeneity between the symptoms, between the patients, and between all the things that is happening. So we think that at the end, these epigenetic layers, adding one more step closer to the complex brain situation and MS progression, and then we will have better treatment in the future.
Okay, great. Yeah, and you already explained how that would happen, that it would be personalized treatment. So can you give us a rough idea of how far or near is this practical use away?
Yeah, I mean, it's a little bit challenging to answer this question because I mean we never know how good this research are going. But the thing is that we know nowadays, like if we compare the situation that we have now in the market of the MS medications, than like 30 years ago or 50 years ago, we have a lot of more in the market. So it means that we have had a lot of progression in the medications and finding the medications. So we think that if we know better this situation, that how these networks are working together, we would actually have this understanding that how we can control better the disease. And I think DNA methylation is one of those. So DNA methylation is one of those factors that can actually help us to understand better this situation, and it would add a lot of important information to this layer. So we are still at the very, very beginning of this road. So I think it's still a very long way to go. But the thing is that it's nowadays we have also a lot of technologies. So sometimes you see that, okay, now there is AI also around. There are a lot of technologies around us. And I think at least for us researchers, we are in a honeymoon somehow, I should say.
That's a nice picture.
It's a honeymoon of research. So you see that a lot of other technologies are around us. There are like very super advanced techniques, super advanced technologies around us. A lot of even people awareness. So now the society is understanding us better than before. And I think all these ones together will help us to go faster and faster towards the new medications for the MS patients.
Yeah. Okay. Yeah. Of course. Yes, it's nice. It's probably like that one movie with Mr. Black or so, where he started as an old man and become younger. So let's say research was like 50 years married and now getting back to the honeymoon.
Exactly.
Because of all the surrounding, yes.
Yes.
Yeah. How might techniques like CRISPR or other epigenome editing tools be used to correct harmful epigenetic changes in MS patients?
Okay, so I think everybody nowadays heard about the CRISPR systems. It's like a system that we can edit the genes. So it means that we can change the genes like in the body. And then somehow some people are scared of this situation that, okay, it can be good or bad. And of course, every technology has this kind of situation. But for us, something that is very important is actually sometimes we don't have any other option. So CRISPR is something that is another tool in our hand that can be very useful. And when I was tailoring back like 20 years back, I think we were using a lot of different small tools actually to make something. But nowadays, those tools in my hands are these CRISPR tools, I think.
Mm-hmm.
It's one of them at least. So we can use them and then they are rapidly actually developing in the field. And we think that it would be very important to find them to find the new network between the genes, so they can help us actually to edit the genes and understand that how these edits actually can help to understand better these networks. But one thing that is always the priority for us is actually to understand the safety of these systems. So we need to be sure that these systems are safe for humans. We are not going to fix one problem and make 10 more problems for the person.
Mm-hmm.
So that's always a situation that we are trying to understand it better. But again, I think the CRISPR system will help us, and it's one of those tools that we think that nowadays is available, that it makes our work much faster than before, as I explained before. But it's not just them even. So we are not even limited just to CRISPR. So I think we have other things around us. And one of the things that I would like to highlight is also these stem cell technologies that nowadays we are using in our research, as I said. So technically, we can take a little bit of the skin of the patient or person and then make a stem cell from them and make a brain on them from the same cells in the dish. And that's a very cool situation that you see that from a piece of skin, you can make a very small brain on the dish in the lab that we call the mini brains. And these mini brains, yeah, they have a very nice term. So these mini brains can help us that we can target those ones first. We can try to test our medications tested first on these kind of models. And at the end, we can go to the situation that we don't want a lot of animals to be killed. We don't want a lot of patients to be suffered, but we just want to test everything first on these models, including the CRISPR, including these millibrain organisms. And then at the end, whatever we found that it makes sense, then we will try it in animal models. And at the end, we treat it as a medication available for MS patients.
Cool. Yeah, very cool. And I'm doing, besides of the multiple sclerosis master's program, I'm doing the UPATI, European Patient Expert, so for patient advocates. And there was CRISPR a lot around when it was about clinical trials. Of course, it was more on cancer treatment, but I think it's really going to an advanced stage where it's not a scary technique, but people know quite a lot what they are doing. So how does these findings open the door to developing treatments that specifically target the brain's processes in progressive MS?
I would like to mention that in the field of MS, we were somehow benefiting from the other autoimmune diseases. This is a fact that patients and even the community should know about it. So it means that when we are talking about the autoimmune disease, it doesn't mean that we are just talking about MS. There are a lot of shared things between the autoimmune diseases, and we can benefit from each other. So when those guys are doing research, for example, in other type of disease, we can get the data from them and we understand, we make sense. We are trying to make sense of those data in terms of MS. So we think that, okay, and that's one of the reasons actually that we have a lot of data available for our MS. So for example, because there are lots of research ongoing on the relapsing remitting forms of MS, because that we have those information from inflammation, and there are some other inflammatory disease that the data is coming from them. But while for the progressive, we cannot link it really that easy to the other diseases because it's not happening exactly the way that we want it. while information could be shared between many different diseases. So we think that even these tools that we are making and this kind of research that we are doing is not just benefiting for MS, but it would benefit for other type of diseases. But at the end, we were thinking that, okay, one day, this type of traditional medications that we do have in the market would change to more specific medications for MS, or as we mentioned, personalized medicine. So I think we are much faster going towards the more precise medications. And I was making jokes of this situation with my colleagues who are medical doctors. I was telling them, soon I will be the medical doctor. You guys should sit somewhere else, because I think that in the future, the traditional medication would change to the more molecular or genetic medications. Then I would be the medical doctor by definition, just like that.
Yeah, making Star Trek live.
Yeah, exactly.
Yeah, doing the scanner, and then you have the medication and everything is fixed. Or there's quite some movies where you are just going into a cabin and then you are refurbished in a way.
Yes, hopefully.
Yeah. Okay, so could the findings in this study help explain why conventional immunotherapies are often less effective in patients with progressive forms of MS?
Yes, and that's, I think we discussed it a little bit. So back to the discussion that we had. So inflammation is the first step of MS. That's happening more in RMS, while the death of the neurons is something that is happening in progressive MS. And in this situation, we need to know better this mechanism. As I said, brain is very well protected, so we cannot have access to that. So we think that one of the main reasons that we don't have enough medication for progressive is this situation that brain has. There are lots of actually trials, a lot of actually experiments has been done to be sure that we can understand better this brain. But even just until recently, we didn't have even a brain map. So it means that the brain map in cellular resolution, it means that which cells are connected to which cells and then how they are performing together, how they are working together, and all these functions. But recently, we have much more of this information. So I think that at least our study showed that there is a link between the blood and the brain, and this is something that has been known for a while, but now we are checking and we are finding more kind of like evidences here and there. And somehow it's a little bit tricky to understand that how a signature that you find in the blood can show something in the brain. So this is something very, very interesting. But nowadays, even we are searching for biomarkers in the blood fluid. So, for example, we are thinking that if there is a specific type of the cell in the brain dying, for example, out of the disease or medications side effects, we can track back those ones in the blood of the patients. Mm-hmm. So we are trying actually to find genetic markers that are released from the MS brain cells to the blood, and then we are measuring those ones and understand, okay, now this process is happening in the brain, and we have this medication to stop this situation, or we can make some changes to stop this situation. So I think that's one of the main things that actually change this situation. But again, back to this situation, we don't know enough about the brain, and that's why we don't have enough medication for progressive form.
Yes, yeah. It's really quite complicated, and I had many interview partners here to discuss it. And for the German podcast, again, I have to bring somebody for the English one. Professor Stadelmann-Nessler, who is doing neuropathology, where she tried to explain how complicated it is. And that's not easy, but of course, working together with all the different research departments, neuropathology, neuroimmunology, let's say, speed ups research outcomes a lot. So you already mentioned a bit the role of environmental factors like smoking, but there are other factors like diet or stress. What role do they play in influencing epigenetic changes, and how can lifestyle adjustments complement medical treatment? Because this is something we can do right now. We don't have to wait for your research outcome.
Yes, exactly. I think this is a question that I get a lot from the audience or from the MS patients when I'm presenting. And we know that as we define the epigenetic is exactly the effect of environmental factors on the genetic factors or genetic background. This is somehow a little bit tricky. As we discussed, epigenetic factors, at least DNA methylation is very sensitive factor. So whatever we do in our life, it's affecting this DNA methylation. And this DNA methylation can affect the gene regulation in our body. And we know that this gene regulation is affecting the outcome of MS. So somehow we know that we cannot select our genetic background. This is what it is. I mean, we get it from our parents as a gift, and we need now to deal with that. To dealing with this situation, we can actually handle the environmental factors. So we know many of the factors that are involved in epigenetic situation and can affect the disease, this MS disease. And one of the most important ones is smoking, for example. We know that the smoking actually makes marks on the genome with the help of epigenetics. It can change the regulation. It can make inflammation, and these inflammatory related genes can affect the outcome of the MS patient. So we always actually try to advise the MS patient not to smoke, to change their lifestyle. A better diet is doing exactly the same thing. Maybe reducing the stress is a key factor that I think is advised by many researchers and doctors, and for MS cases, and also physical activity. I mean, all these kind of factors can have positive and negative effects on the DNA methylation and on the gene expression, and at the end on the symptoms of the MS patient. So we recommend always actually to control or to handle these things together to be sure that, okay, as you said, why we cannot change the genetics, now it's time actually to control these environment factors without waiting for our research, of course.
Yes. Be the DJ of your own life, in a way.
Yes.
Yeah. Okay. Great, Majid.
Yeah.
Okay, let's come to the quickfire Q&A session. Complete the sentence. For me, multiple sclerosis is?
I think multiple sclerosis is a condition that, while it is still challenging, but is no longer as severe as it was before.
Absolutely.
And with the rapid progress in research and new treatment strategies, I believe that we will hear very good news in near future.
Yeah, yeah. And I will always try to bring them to the audience, to our community, from ACTRIMS and from interviews like with you and other colleagues of yours. So what development would you like to see in the field of multiple sclerosis in the next 5 years?
To me, it's a very simple, and it is something that I always think about, like any medication to control MS progression.
Yeah.
That's something that I would love to see.
Yeah, absolutely. Okay, so finally, what message of hope or encouragement would you like to share with the individuals living with MS?
I would like to tell them, don't give up. I mean, we know that it is a very tough situation. Some days it's really tough, but please remember that researchers around the world, including myself, are working hard to find better treatments and solutions for you. And I truly believe that one day we will hear in the news that MS is cured. And now we researchers need to open a new research field and working in another disease. That's something, a dream that I think it will come true soon. I want to hear that news, that MS is cured.
Yeah. Yeah. So I don't want to retire in a way, because I want to write 100 books and a lot of children's books as well. But let's say I would have no problem with shutting down the podcast, the German and the English one, because it's cured and everybody who had the disease could be healed and could be helped with all the symptoms management. Fantastic. Would have no problem moving back to the children's books.
Sure.
Would be lovely. And we could have a green tea on that, because I'm not drinking alcohol, or orange juice or something like that.
Yeah, I will go back to saffron culturing, or even maybe to tailoring. It's fine. I have my second job.
Yes, you have an alternative. So how and where can interested people follow your research activities, Majid?
Yeah, I mean, normally we are present in the big conferences like ACTRIMS, as you mentioned. And also I'm trying somehow like to somehow make it simple text and then share it in my LinkedIn profile, that if they just search me by my name, they would find me. And also we are sharing a lot of information in our research group that you can find in the Karolinska Institute website, where I need to mention that I'm part of the group that is led by Professor Maja Golic. So our whole team actually working on this multiple sclerosis epigenetics and the link of environmental factors with genetics.
Fantastic. And of course, I will put these links into the show notes and into the blog article so you can find it there. And you don't have to use Google, but you can, of course, always, my dear audience. So Majid, thank you. It was a pleasure to hear about your research, and thank you for all these easy to understand explanations, and hope to meet you at one of the upcoming ACTRIMS. And please go on with all the research and say thank you to the team as well, because I know it's always a combined research. Yeah, so thanks a lot.
Thank you so much for having me. And it was a great pleasure to talk to you. And I hope that your audience also will benefit from this discussion that we had just now. And hope to see you soon.
Yeah, thanks. Bye-bye.
Bye.
I hope you enjoyed the interview and learned something new. For me, it was definitely a very interesting topic, and I hope to see further advancements in the future so that there are new treatment options. By the way, next time I will have an interview on leveraging artificial intelligence in multiple sclerosis care and shared decision-making with Dr. Stefan Ebner. Dr. Stefan Ebner is a keynote speaker, an official Google spokesperson, a machine learning expert, an author, data lover, university lecturer, and lives in Bergisch Gladbach in Germany. So this will be an interesting one again, and I hope you tune in. I hope you stay as active and positive as possible. And please rate the podcast wherever you are listening to it and recommend it to other people who might be interested in it. And so, see you next time. Bye. Thank you for listening today. I hope you found it helpful. You may also be interested in the previously published episodes and posts, which you can find at ms-perspektive.com/podcast. Perspective with a K. You can also get a free PDF with 11 tips for your positive impact on MS at ms-perspektive.com/newsletter. If you'd like to get in touch, you can reach me at nele@ms-perspektive.com or on social media. See you next time.